All right, so today we're going to start Chapter 8. So Chapter 7 was photosynthesis, so that's how plants are making carbohydrates. Chapter 8 is how we're going to be processing carbohydrates, which is similar but really the opposite direction. So the goals for this chapter are, one, to be able to describe what cellular respiration is, which we're going to get into today, do a brief overview of. We're going to understand how reduction oxidations work and how they can be a form of energy storage and energy transfer. We're going to be able to understand and describe the 4 phases of cellular respiration, which again, that's the process through which animal cells are going to kind of digest and process carbohydrates and be able to differentiate between aerobic and anaerobic respiration. So does anyone know maybe what these terms mean, aerobic and anaerobic? Yeah, so aerobic is with oxygen, anaerobic is without oxygen. So some of the phases of cellular respiration require oxygen and some don't. And so we'll get to see kind of the decisions that cells make as to which route they go down at different times. So cellular respiration, again, this is sort of like the opposite of photosynthesis. In photosynthesis, plants were absorbing energy in the form of solar energy, and they were creating carbohydrates which have a high chemical potential energy. In cellular respiration, animals are going to be breaking down these, they call them sugar molecules, carbohydrates, glucose. These are all kind of acceptable terms. They break down these molecules, and they're going to be using that to do useful work, like to move around, to jump, to talk, to lecture, whatever. And so animals are going to be releasing carbon dioxide and taking in oxygen, whereas plants did the opposite. They take in carbon dioxide and release oxygen. So we can see that these cycles are really intertwined. And so what you learned about photosynthesis is going to help you hopefully a little bit understand cellular respiration. So the kind of overall reaction for cellular respiration is we have glucose and oxygen coming in, and we're exhaling carbon dioxide, releasing water, and we're producing energy as well. So that's the important thing really, is we have energy in the glucose that we eat in the chemical potential of the glucose in those bonds that we can release to do other work. Lots of different examples. And so eating is how we get our glucose, and breathing is how we get our oxygen. And this is why we have to eat and breathe, is so that we can power this cycle. So it's like really core to what makes animals function, this process. And it drives a lot of their behavior. So eating, breathing, we excrete water as waste, we exhale carbon dioxide. These are all dependent on cellular respiration, and they kind of describe a lot of what we do as organisms. So one way to think about it is we're going to be using ATP as a molecule a lot. So glucose is like a big chunk of coal, like a big bulky energy storage molecule. And we want to break it down into smaller, more useful, portable things like ATP, which are kind of like charged batteries. So we take a big chunk of coal and we kind of process it, and then we can have batteries that are more portable, and you don't need a little furnace on your cell phone, so you can just put in batteries. It's similar to like what cellular respiration is doing. We're taking a big, bulky energy storage molecule like glucose, and we're making it into smaller, more kind of bite-sized, portable energy molecules like ATP, which are going to be easier to use in different reactions. So producing ATP, is that going to be exergonic or endergonic? Producing ATP will be endergonic. What about breaking down glucose? For breaking down a big molecule like glucose into smaller molecules, is that going to be consuming or releasing energy? If it's coupled with an endergonic reaction, what does it have to be? Exergonic, yeah. So we're going to be breaking down and releasing energy from this glucose and putting that energy into making ATP, which is the energy that we'll be talking about. So this is the overview, and now we're going to get into kind of the finer details. But does everyone sort of understand the importance of cellular respiration? Does everyone already think this is the coolest chapter in the book?
Yeah.
Great. All right. So first we're going to talk about these oxidation reduction reactions, or reduction oxidation. Either order is fine. So just to get some vocab first, then we'll talk about how they're used. A nice little mnemonic, I wish Margaret was here, she's always asking for mnemonics, is oil rig. Oxidation is loss, and we're talking about electrons. So oxidation is the loss of electrons, reduction is the gain of electrons. So we have our example over here. We have molecule X is giving an electron to molecule Y. So X is losing an electron, so that would be loss of electrons, that would be oxidation. And Y is gaining an electron, that would be reduction. So Y is being reduced by X, X is being oxidized by Y. And so these happen in pairs because the electron is going from one to the other. So that's the vocab for which is being reduced, which is being oxidized. And now we're going to talk about how those can store energy. But does everyone understand for now that this is losing an electron, and therefore it's being oxidized, and this is gaining an electron, it's being reduced. So if we add an electron to it, it's like we're adding energy to it. So if we think of it like batteries, when you put electrons into a battery, you know, when you plug it into an outlet, as though that's how batteries work. But anyway, when you charge a battery, you're putting electrons into it, more or less, and you're charging it up. So is that going to be being reduced or oxidized? If it's gaining electrons. Yeah, reduced. Great. So we'll keep working on the vocab to kind of make it more concrete. But and then we have a full battery, and we're putting it into some sort of toy, and the battery eventually becomes empty. Is the battery now being reduced or oxidized? Oxidized. And so we put energy into it, and now we're letting energy come out of it. And so there's a lot of molecules there's a few molecules that we'll be talking about in this chapter where we add electrons to them to make them more energetic.
Is it called reduced because when you're putting electrons into it, it's becoming more negative?
No. Well, why is it called being reduced?
That's the last slide, right?
Well, I know that's what's happening when it's being reduced. Oh, I can't go backwards. I don't know why it disappeared. What? Is this not clear? How would you like to present it? Maybe we'll talk about that. It can be a big group exercise. How should I present information so I don't give you headaches? Maybe I'll go through it and then we can make suggestions. So instead of batteries, because our cells can't use batteries, that would be awesome if they could, they have to use NAD and FAD. So NAD, we're going to add electrons to, and a hydrogen to make NADH. FAD, we add electrons to make FADH2, and also the hydrogen. But we're not going to really talk about these hydrogens as much. We're mainly going to focus on where the electrons are going, because it's going to make the whole analysis a little bit easier. So if we're adding electrons to NAD, is it being reduced or oxidized? Reduced. You guys are so good at this. You teach me. So we have NAD is like a lower energy form. We add electrons to it, and now it has more energy, and it's NADH. So it's more unstable. It has a higher energy, and it's looking to get rid of those electrons. And when it gets rid of them, that's going to be an exergonic reaction. To add the electrons onto the NADH is endergonic. So now we can couple the breakdown of NADH later into NAD. It can be part of a coupled reaction to drive another endergonic reaction. So the point of a lot of the steps in cellular respiration and also in photosynthesis was to change the form of the energy into something that's usable in reactions. And so glucose was really too big for us to work with. It had too much energy. It wouldn't have been easy to make ATP from the glucose. But we can break steadily through a series of reactions that glucose down into NADH and FADH2, and then use those to make a lot more ATP. So that's kind of the roadmap of what we'll be talking about. We'll be talking about how we are going to make NADH and FADH2 from the glucose. So we're going to add electrons to them, and they're going to be reduced. And then later down the line, they can be oxidized to make the ATP. You want to get into the details now of cellular respiration? Are you excited? I would take sarcastic answers. That is fine. Any answers are fine at all. Again, the objective of cellular respiration is to break down glucose and make ATP. And we're going to do that in 4 steps, 4 distinct steps that are happening in different locations and have different purposes. So first, we're going to have glycolysis. So I'll go into the specifics as we go through the next slides, but just to give you an overview of what's happening. First, we have glycolysis. That's making some ATP. It's happening, out in the cytosol, sometimes far away from the mitochondria. Again, mitochondria is where most of the action is going to be happening. We have the preparatory reaction, where we're going to take some of the products from glycolysis, and we're going to move those into, so we have glycolysis here. We're going to move those into the mitochondria, and we're going to be making some of this NADH and ATP. We have the citric acid cycle, where we're going to break down the rest of our carbon. So after this, we'll have no more carbon in our steps, and we'll make more NADH and FADH2. And then the final step, the electron transport chain, is going to be sort of similar to what we talked about in the light reactions in photosynthesis, where we're going to let these electrons flow down from really high energy molecules like NADH and FADH2, and they're going to flow down this electron transport chain, and as they go, we're going to be making ATP. And this is where most of the ATP are made. So from each ATP molecule, we're going to be making on the order of like 36 to 38, from each glucose molecule, we're making on the order of 36 to 38 ATPs. So don't get too bogged down in like the exact numbers. I'm not going to ask you how many ATP for each glucose, but I think what's important to know is kind of like the scale. We get a lot of ATP molecules from one glucose. And so when we break it down into these kind of like smaller bite-sized molecules, it's more on the order of like what different endergonic reactions would require to happen. So now we can just go spend 1 ATP to make another biomolecule down the road when we need to do that. Whereas before, if we just had glucose, we would have wasted a lot of energy because we'd have like a $100 bill to buy like a $1 thing and just lose the change. Whereas now we'll have smaller energy currencies in the form of ATP to make all of these reactions happen. So do you think in all these steps there are any enzymes involved?
Yep.
What do you think those enzymes are going to be doing? Speeding it up. Yeah, what would happen if there are no enzymes? Yeah, and what would happen to the organism, the cell? It would die. That's why enzymes are so cool and exciting, just like everything else in biology. Okay, so here's a little more detail, and we're going to get into each of these steps one by one. Glycolysis, we're breaking down the glucose. What? Do you not like my slides? I'm really offended.
No, it's awesome. Can I have some color?
Yes.
It's a tough crowd tonight.
Color. Awesome. Okay. Makes it so much better.
Great. Now we're all engaged. Yeah, because there's color involved. Okay. I can add color in future slides. Okay. Noted. So glycolysis, our first step, that's where we're breaking down our glucose. So we have glucose, which is, again, 6 carbons. I'm holding 7 fingers, 6 carbons. And we break it down into 2 molecules that both have 3 carbons. So we're going to break down this glucose. In the process, we're going to make ATP and NADH. This NADH, again, this is going to store energy for us to use later to make more ATP. So it's really, this NADH and also this FADH2 are really only going to be useful in cellular respiration to make ATP, but in the end, all we care about is the amount of ATP that we make. So glycolysis, we're going to be breaking down our glucose. We're going to break it in half. We're not going to really lose any carbons, but we're just going to break them all the molecules in half. We're going to make some ATP and NADH. Again, the numbers aren't really important, but just know it's a relatively small number here. Then we're going to send that pyruvate to the next reaction. So this is going to happen in the mitochondria, in the mitochondrial matrix is what it's called. So inside the cell, we have the cytosol, which is like the, does anyone remember what the cytosol was? How would we describe it?
cytoplasm.
Yeah, great. Yeah, those are both synonymous. So, what's the cytoplasm? Yeah, sorry. So, right, it's this like semi-fluid that fills the cell. It's this really viscous fluid. It's water-based, but it's just jam-packed with different biomolecules, and it's so full of those that it's this really viscous, like maple syrupy type semi-fluid is what they call it. So, glycolysis is happening out in the cytoplasm or the cytosol. Sorry, I guess I should have stuck with cytoplasm. Keep my nomenclature consistent. So glycolysis happens in the cytoplasm. Preparatory reaction is going to happen in the mitochondrial matrix, which is like the cytoplasm of the mitochondria, you could think of. And here we're going to break down the pyruvate, which is 2 molecules of 3 carbons each. We're going to break it down into 2 molecules of 2 carbons each, and then we're going to lose 2 of those CO2 carbons to CO2, which we're just going to exhale. And in this process, we're just going to make some NADH. So no ATP are being made in this step. Third step, we take the 2 carbon molecule from before. We have 2 of those. We're going to break them down into carbon dioxide, which we're just going to exhale. So now after this step, we have no more carbon left. We've broken down all of our glucose into CO2. And now we're left with NADH, FADH2, and ATP. So again, we said before that we're going to make like on the order of like almost 40 ATP, but in these first 3 steps we made 4. So we still have a lot to do in this last step, but what we do have is all of these NADH and FADH2, and we're going to use the energy that was stored in those molecules to make our ATP. So these are the 4 steps. We can see that the last step really takes these high energy molecules in the form of NADH and FADH2. It takes those molecules and it uses those high energy electrons. It feeds them into the electron transport chain and it makes the rest of our ATP. So the first 3 steps are really about breaking down the glucose into carbon dioxide to create some energy in the form of, or convert some energy, I should say, to NADH and FADH2, and then the last step we use all of those high energy molecules to make the rest of our ATP.
So basically you just taking some from one step and just moving it down to the next step.
Yeah. And as we go, I'll write it on the board where all the carbon and where all the energy is going, so that could help us keep track.
Hold on.
All right.
Is this going to be like you want to
What I write on the board is all that will be expected for the exam, but I can also post it online.
So is the citric acid cycle Krebs?
Yeah. Sorry, everything has like 20 names to it. It's difficult to choose. Can I go ahead, or are you still? Great. Okay, glycolysis. So again, glycolysis, we're breaking down a 6 carbon molecule in the form of glucose into 2 3 carbon molecules, and in the process we're making some ATP and some NADH. So this doesn't require any oxygen. It's taking place in the cytoplasm, which is outside of the mitochondria, and it has a lot of extra steps that aren't shown here. And so you could spend a lot of time studying just glycolysis, as many people probably have, and there'd still be a ton to learn. But I think the overarching concepts are what's important. And so I'm going to try to write where all the carbon and where all the energy is going in each step, and that's really all that we need to remember for the exam. But I know that you'll just naturally be so curious that you'll study more than that.
Like, yeah.
Or at least if anyone asks you what this class inspired, you would say to study more. So you don't have to remember the exact number of ATP and NADH that are made, but I think you should know that when we break down one 6-carbon molecule, we have to make 2 3-carbon molecules. So this is glycolysis. This is where the carbon is going, and this is where the energy is going. So we had some energy that was in glucose that we kind of siphoned off to make this ATP and NADH that won't be in the pyruvate. And also we have some energy that we just lose to heat because that's one of the laws of thermodynamics. Which law is that? Second law. Every reaction we have, we're losing some energy to heat or entropy or whatever you like to call it, disorder. So, are we okay? Yeah. Okay. So there's 2 again. Only things that you really have to know for the exam to describe each of the steps are going to be what's on the board. But I think if I just wrote what's on the board straight down, then it wouldn't really be a very compelling story. So I'm going to give some extra detail about what's happening. But if you're feeling like you're falling behind, just focus on what's being written on the board. So there's 2 steps in glycolysis. There's an energy investment step where we have to actually use 2 ATP. And there's an energy harvesting step where we're going to kind of gain back 4 ATP and also 2 NADH. So in total, we're only making 2 ATP, but we have to first kind of spend some money. And the way we're spending that money is or spending that energy is in these enzymes to kind of like prime them to get them ready to do the reaction. So to get everything going, kind of get the momentum going, you have to use ATP to get that happening. So as you're coupling these reactions, you actually have to use ATP to make this reaction start moving. Otherwise, the activation energy will be kind of just insurmountable. So you have the activation energy to get through the transition states as you break down the glucose. And without the energy that's being released during this energy investment step, you wouldn't ever get over that activation energy. So you have to first spend 2 ATP, and then after you've gotten over that hump, that activation energy, you can then harvest the energy that you released as you broke down the glucose to pyruvate. And as you harvest that energy, you're going to be able to make 4 ATP and 2 NADH. And again, this is happening outside of the mitochondria, just in the cytoplasm. So, many steps to this. Again, please don't study this thinking it'll be on the exam, but if you feel like studying it, please be my guest. So we have glucose up here. We're adding in 2 ATP, as you can see here. We're making a G3P. Has anyone seen that term before?
Yeah.
Look at all this color I have on this slide. Aren't you guys happy?
Yeah, it's awesome.
So
Pastels.
So love it.
So G3P, this is the same molecule that plants made at the end of this the Calvin cycle. And then as we break this down further into pyruvate, we can make more ATP and NADH. But again, we had this initial energy investment step to break it down, and then we could break it down further and harvest the energy from that. And we get that energy from the potential energy, the chemical potential energy in the bonds of glucose. And then G3P, all the way down to pyruvate. So which has more energy, 2 pyruvates or 1 glucose?
They're the same.
So first of all, we know there's been a series of reactions to get from glucose to 2 pyruvate. So what happens at each time we have a reaction? We lose some. So that alone should tell us that 2 pyruvates have less energy than glucose. But also from this glucose, we harvested enough energy to make some ATP and NADH, so we lose even more energy to that. So 2 pyruvates have a lot less energy than 1 glucose. And as we go through each step in this process, each of the glucose molecules is going to have less energy than it did before. And we're just going to slowly take energy from it to make the ATP and NADH. So, little review. What does glycolysis mean? I had a little description in quotes before. What did it say?
Break down
Oh my gosh. You're making me so happy. What molecules do we start with? Glucose. Yeah, mainly just glucose. Pyruvate and don't read it off the board. Just read it out in your mind. And we end with pyruvate, ATP, and NADH. Yeah. So we start with glucose, and we end with pyruvate, ATP, and NADH. And where is this happening? Is this happening in the cytoplasm? Yeah.
Outside.
That's where the rest of the steps will be happening. So you're close. Great. We ready to move on to the next step?
Well, we need to know if each step requires oxygen.
No. That's a good question though.
Anaerobic?
I should have thought of that question beforehand, and I could have made it more clear for each step, but I didn't. But good question. So just to go over the little overview we had before, this is sort of like colored, bold and gray.
Look at the light color.
Yeah, so I'm moving in the right direction. I could write my name again if you want.
But the light also.
Oh yeah?
Gotta pass the lights on.
Yeah.
Is this what I've been missing off that?
Thank you.
Oh, that's
Is that worse?
It's all cemented to strain off.
Oh, I'll just keep shouting. You won't fall asleep, don't worry. So there's 4 steps. We just went over the first of the 4 steps. So now we're going to take some of the products from the first step and feed them into the second step. So now we have the preparatory reaction. So here we're going to take the pyruvate from glycolysis, and we're going to break it down into this acetyl-CoA. So acetyl-CoA has 2 carbons, pyruvate has 3 carbons. Where does the third carbon go? CO2. And what happens to that CO2? It's just like a waste product to us. We just exhale it. And we don't want too much of it in our blood because that's going to change our blood pH. So we have to get rid of it. It's actually like sort of a toxic molecule in really high concentrations. So we have to get rid of that CO2. And we're going to be making some NADH as we go from pyruvate to acetyl-CoA. But we're also going to be moving from the cytoplasm to it should say mitochondrial matrix, but instead it just says mitochondrial matrix. Sorry, I got cut off. But we go from the cytoplasm and we're going into the mitochondria. And so as we move into the mitochondria, we're going to be going through this preparatory reaction. Right on the board. Alright, so the preparatory reaction.
Is the mitochondrial matrix also included?
Yeah, it is. Yeah. But it's also different than the cytoplasm. It has a different makeup of biomolecules. So it'll have like different enzymes in there. You probably will have a lot less lipids. But so the point I want to make though is that the mitochondria is separated by a membrane because its composition is very different than the cytoplasm. And again, this is why eukaryotic cells are capable of much more complex behaviors because they can have these separated compartments to do very separated and specific tasks. Again, though, to overview, we're taking the pyruvate that we ended with in glycolysis, and we're feeding it into this preparatory reaction. So it's going to go from the cytoplasm into the mitochondria, and we're going to release 2 carbons as CO2, and we're going to make 2 acetyl-CoAs, and these both have 2 carbons in them. So we had 6 carbons here, 6 here, 6 carbons here, and 6 carbons here. But do you think we're going to have 6 carbons to start the next step? No, because we're going to lose this CO2 as waste. So with the CO2, one way to think about it is we've taken all of the energy from it that we can. Now it's at its kind of least valuable form. So we're just going to get rid of it because we can't do anything with it anymore. Do you want to finish the 4 steps and take a break, or do you want to take a break now and then finish them? Keep going. Oh yeah, we can try it. So what's the point of the preparatory reaction, or what's happening in the preparatory reaction? Great. And what else are we doing?
We're making NADH.
Making NADH, and we're going from the cytoplasm into the mitochondria. Those are all correct answers. And so where is this occurring?
In the mitochondrial matrix.
Great. And so,
Wait, can you explain what it is again?
yeah. So in the preparatory reaction, we're breaking down our pyruvate into CO2 and acetyl-CoA. We're making some NADH in the process, and we're moving from the cytoplasm into the mitochondrial matrix. Oh, I kind of want to add a third column with location where this occurs. I think that's fair. I'm going to add a third column.
There's no room. Put on the left side. I mean,
I can move. I can move.
Oh, God. Yes, it did. It did. I'm sorry.
All right.
So these are two different columns.
Yeah. So the location, what's happening to the carbon, and what's happening to the energy.
So all of this is going in.
Yeah. So these are sorted out like that, but I think we can. I have faith in you guys.
Will you leave all that up until after class anyway?
Absolutely. Okay, so now we've gone through the first 2 steps, so we're like halfway there. But even though we're halfway there, we've made very little ATP. We've only made 2 ATP so far, and we've made 4 NADH molecules. So far we haven't really done as much ATP production as we might have thought, because again, the ATP production is mainly going to happen in this final step. The point of the first 3 steps is to make the NADH and FADH2 that feed into the final step. So we have made 4 NADH, and we've made this acetyl-CoA as well. So that's what we have going to our 3rd step. Yeah, I didn't want to put the numbers up there because I think it's extra info. That's not. Is that more confusing though to not put the numbers? Oh, okay. So 3rd step is the citric acid cycle. So there are many steps to this. Again, we're not going to go into each step because I just think that's just going to be kind of tedious and not really helpful to you guys. But the important thing to realize, in my opinion, is that we have 4 carbons going into this step in the form of 2 acetyl-CoAs. And at the end of this step, we're going to have 4 CO2s. So again, CO2 is like the least valuable form of carbon in the perspective of cellular respiration. Acetyl-CoA has a lot more energy in it. So we're going to take the rest of the energy from the acetyl-CoA and we're going to put it, or take it all out and put it in the form of FADH2, NADH, and ATP, and the carbons are just going to leave as CO2 as a waste product.
So we create 6 NADH from it, 2 FADH, 4 CO2, and 2 ATP.
Correct. Yeah. But again, it's important to realize that we had a lot of energy in glucose, and we're breaking it down over the course of cell respiration into 6 CO2s, which have a lot less energy. And all of that energy to this point is going to go into making NADH, FADH2, and ATP. So we had chemical potential energy, in our glucose. We're going to make these FADH2 and NADH by adding these high energy electrons to them. So if we're adding high energy electrons, are we reducing or oxidizing them? Reducing them. And so what's going to happen later down the road to these molecules? Will they be oxidized or reduced? Oxidized. Great. And when they are oxidized, that's how we're going to be making the ATP. But again, so this is happening in the matrix of the mitochondria. We don't fall.
So like switch this citric acid.
Yeah. Incorrectly. So just to review, we have our 4 carbons going into this in the form of 2 acetyl-CoA molecules, and they're going to leave as 4 CO2. So we're going to take all the energy we can out of these, and we're going to make from those acetyl-CoA as they go into CO2, we're going to make NADH, FADH2, and ATP. So this is like one big coupled reaction, where this is the exergonic reaction, and making these is the endergonic reaction. And the endergonic reaction is the act of reducing these molecules, adding electrons to them. So now we have energy that we've stored in these molecules that we can move to the final step. Do you have a question?
I'm not sure.
Okay, yeah, you can ask later if it comes up. So this is the third step. Again, so we're beginning with these 2 carbon molecules, and well, I don't know if we really want to get into this, but I just this won't be on the exam, but just so we can see the comparison to photosynthesis, because I think it's interesting to see how very different sets of organisms can have really similar reactions and similar ways of doing things. So we're going to add our acetyl-CoA to a 4 carbon molecule. It's going to go through around a cycle, and we're going to regenerate this 4 carbon molecule. We're going to lose some carbons as CO2, and we're going to make these high energy molecules out of it. So it's sort of like in some ways the reverse of the Calvin cycle. Instead of putting energy in, we're taking energy out. Instead of adding CO2, we're losing CO2. Can we see like the similarities and the differences between the two? So they both are kind of interested in conserving the reactants that they'll start with. So we want to make sure at the end of the cycle we're reproducing this 4 carbon molecule, just like the last step of the Calvin cycle was to regenerate our 5 carbon molecule. Because these all have to be sustainable, because they're happening so many times, probably like on the order of like once a second or something like that. They're happening very frequently, that if we were to not make it sustainable, it would be very expensive to do that. So it has to be sustainable. You have to regenerate what you started with. So we see a lot of cycles in biology like this. But the difference from the Calvin cycle is that rather than taking in CO2, we're releasing it, and rather than using high energy molecules in the form of NADP, pH, like in photosynthesis, we're creating NADH and FADH2 and ATP. So it's similar, but sort of reverse. Which I think we all agree is a thrilling concept. Maybe just
My brain is crying.
like joy. I've decided I'm just going to be super optimistic for the rest of the semester.
All of this energy you said is coming from the bonds.
Yeah.
The carbon,
In the glucose, yeah.
and that's why the carbon is leaves after the bond is broken.
Sorry, say that one more time.
And so when the bonds are broken and it takes the energy, the carbon just leaves.
Yeah, right, yeah. So carbon by itself won't fulfill its octet rule, so you couldn't just float it out on its own. It's going to combine with other oxygens to just fulfill its octet to be a little more stable, but it still has much less energy than it did in CO2, in glucose. So glucose has a lot more energy than 6 carbon dioxide molecules. And that's the energy that we're going to be using to make ATP.
That's the solar energy? Or
Yeah. I mean, because in photosynthesis we went from CO2 to glucose, and that energy was supplied by solar energy. And then in cellular respiration, we take that energy and we break it down to make ATP. But in some sense, yeah, it was the solar energy. Good question. Isn't that pretty cool that when we eat carbon... You can't say yeah until I finish the statement. And I know you're lying to me. But anyway, it is really cool. So what do we start with in the citric acid cycle? Without
2-acetyl-CoA.
Yeah, 2-acetyl-CoA. Great, good job.
On that.
And what do we end with? 4 CO2s and what else?
2 FADHs, then
So NADH, FADH2, and ATP is what we end with. And so where does this cycle occur? Where is this happening?
in the matrix.
Mitochondrial matrix. And why do we call it a cycle? Why is it citric acid cycle?
Because it keeps on going.
Yeah, but how it keeps on going because in the end you regenerate what you started with.
Oh.
But yeah. Oh, I mean it keeps, but it's to go further into it.
When you say regenerated what you started with, like what
Yeah.
exactly is being regenerated?
Yeah.
Like,
So.
Is it the 4 carbons?
Yeah. Exactly. Yeah.
Okay, so just the 4 carbons.
Right.
We're talking about comparable to the acetyl-CoA that you started.
Correct.
Okay.
Yeah. So you have the acetyl-CoA that you add to the
It's the carbons that are regenerated.
Exactly. Yeah. You regenerate the carbon-based molecules that you started with. Would be a better way to phrase it. All right, we're on to the last step almost.
Yay.
I can live with sarcastic cheering. That's better than silence.
No, my God.
We're really excited.
It's substantially better than silence. I feel like I'm not going as insane when people cheer. So we have in citric acid cycle, we started with acetyl-CoA, and then that was all of the carbon that we had, and we release it as CO2, which is just a waste product in this process. And from that, we're going to harvest some energy in the form of NADH, FADH2, and ATP. So now, going to our last step, we're going to take all of the NADH and FADH2 that we made in the previous 3 steps, and we're going to make that into ATP.
All right.
What?
32, 34, 18.
Yeah.
All the CO2 that's made throughout all these processes is just expel the cellular waste, right?
Yeah, exactly. Great. Yeah. So it's so it'll go into your bloodstream and then to your lungs, and you'll exhale it. All right. So again, we're going to be talking about proteins, which are very exciting. And so these proteins are along the membrane of the matrix of the mitochondria. So we remember from previous lectures and labs that the membranes aren't just phospholipids that seal off the organelle or the cell. They also have embedded in them proteins and signaling molecules. And so in this case, we have proteins that are embedded in the mitochondrial membrane that can facilitate the transition of energy from NADH and FADH2 into ATP. So another term for the electron transport chain is oxidative phosphorylation. So let's break that down into the two words. So oxidative, so we're oxidizing things. So if things are being oxidized, what's happening to the electrons? They're losing electrons. So before we had been reducing molecules to add electrons to them. Now we're going to be losing electrons in oxidative phosphorylation. So NADH and FADH2 are going to be oxidized as they lose their electrons to begin the electron transport chain step. So that's the oxidative part. Phosphorylation all that means is adding a phosphate to something. In this case, we're adding a phosphate to ADP to make ATP. So oxidative phosphorylation really tells you what's happening in this step. We're oxidizing something to make ATP. What we're oxidizing is the high energy molecules that we made in the previous 3 steps to make our ATP. So if I asked you to explain why it's called oxidative phosphorylation, would you be able to do it?
Yeah.
Okay.
So you're losing NADH and FADH on ATP.
Exactly, yeah. And you made those in the previous 3 steps.
So you gonna write it on the board?
Yeah, once I get through the rest of the details, I will, but I will put it on the board, I promise you. It is all in your textbook, if you wanted to read it. But, so, electron transport chain, we're starting at the top at a very high energy level with this NADH losing its electron to become NAD. So, like I said before, this is a really high energy molecule. We added an electron to it, and it really didn't want it, and it's excited to get rid of it. And so when it gets rid of it, it's going to be releasing energy. So we start at the very top with this high energy electron, and it's going to flow down through the electron transport chain through a series of proteins until it reaches oxygen. So oxygen, we know, is very electronegative from, I think, chapter 2. We talked about that. So oxygen is very electronegative, so it's kind of pulling the electron down from NADH through the electron transport chain to get to the oxygen. And as it goes between each protein, we can see it goes a little bit lower on the energy level. So each step, it's releasing some energy. And FADH2 has a really similar process, but we don't have a chart for that. But what's important to realize is that this electron, as it goes down the electron transport chain, is going to be releasing energy, which is exergonic. So now we can pair it with an endergonic reaction. And we know the final endergonic reaction that we want is to make ATP, but unfortunately it's not that straightforward. But this is the first half to the electron transport chain, is the electrons flowing down the electron transport chain from a very high energy molecule that doesn't want the electron all the way down to oxygen to produce water. So we won't really get into the chemistry of why adding an electron here is necessary to make water, but we're just kind of balancing the charges. And oxygen is what we start with when we breathe in, and water is what we're going to be releasing. So, does everyone sort of understand the concept behind this graph?
Yeah. Yeah, it's the molecule, and it's moving down. And as it's moving down, it turns into the oxygen.
Yeah, so we have a high energy electron. Yeah. So we're oxidizing this NADH, so it's going to lose an electron. And as that high energy electron flows down the electron transport chain, it's going to be releasing some energy at each step. And the final electron acceptor is this oxygen. So now we're going to see what happens to that energy. Well, maybe I skipped the slide, but soon we will see what happens to that energy. So this is all happening in the intermembrane space. So the intermembrane space is this area right between the outer membrane and the inner membrane. So this is the intermembrane space. So since the mitochondria has these projections, the intermembrane space is much larger than it would be otherwise. So the amount of volume we have there makes this reaction happen at a much higher rate. So the reason I keep bringing that up is because I think it's interesting to see that the structure that cells use to make different biomolecules or organelles isn't just some random decision. It's done to make them more efficient, to make them more optimized. So it gives the cell the best chance to use the resources it has to do useful things so it can survive. Oh, sorry, down here. So as the electrons move down the chain, the electron transport chain, it's coupled with the pumping of hydrogens up their concentration gradient, which is what we saw in photosynthesis as well. So as the electron moves down that electron transport chain that we saw last slide, it's going to be pumping hydrogens against their concentration gradient. So is this an active or a passive process, moving hydrogens up their concentration gradient? Active. So when those will eventually flow down their concentration gradient, will that be consuming or releasing energy? Releasing energy. It's like water flowing down a hill. Things going down the gradient are generally going to be releasing energy. And so now that we have this concentration gradient of protons, we can let them flow through this enzyme called ATP synthase to make ATP from ADP in a phosphate. So we made all of these NADH and FADH2, so we'd have these high energy electrons that could flow down the electron transport chain to pump protons up their concentration gradient, so those could then flow through an ATP synthase to make ATP. So we began the electron transport chain with 10 NADH and 2 FADH2, and those have really high energy electrons, and those molecules gave their electrons to the top of the electron transport chain. They flowed down the electron transport chain, and then at the end we made ATP by using this ATP synthase. So that's the final step of cellular respiration. Not the end of the chapter, but the final step. Any questions about that? Would you call it the most exciting step? Or the least exciting step? No, no one cares.
I need a break. It's break time now. Oh, well, that's
I feel like an electron right here.
So what carbon molecules are involved in this step?
losing energy.
Which?
I said I feel like an electron right here.
Why? Is you being so negative? What carbon molecules are involved?
That was a good one. Alright.
What carbon molecules are involved in this step?
Are you so quick because you only eat vegetables?
Is that a pun?
It's not a fun.
NADH.
Yeah, but does it have carbon in it? No. So there are no carbon-based molecules consumed or produced in this step. Do we make any NADH or FADH2?
Yeah.
Do we make it in this step?
No.
No. What do we make?
ATP.
ATP. Oh, that's terrible.
So what is consumed? Nothing is consumed?
What do you think? What's at the top of the electron transport chain?
Electron.
Well, that's fair. Where was that electron before?
In this.
NADH and FADH2 are consumed. So all of the NADH and FADH2 from these three steps is being consumed by this final step. I guess I could really, maybe that would be a more correct way to write this. So we take the energy from these molecules and we use it to make ATP. That's probably an easier way to look at it.
No carbons.
Correct. No carbons are involved in this step. All right. Why don't we answer these questions and then we can call it a day a little bit early because we had such a productive class. This actually was really productive. I thought this would take a lot longer. Would it help if I posted this online?
Yes.
I can do that. I can do that too.
Do something
No, just smiling was enough. What do we start with in the electron transport chain?
We start with FADH2 and NADH.
Great, exactly. And so what are those molecules again? What are they doing in this whole process?
They're high energy.
A high energy, exactly. They have high energy electrons put in them. So if you add an electron and they become very high energy, do you think they're very electronegative? No, so this is kind of we didn't really cover this, but they're not very electronegative. They don't want that electron, but we kind of forced it on them, and so they're excited to get rid of it, and they're going to release energy when they do it. Oxygen, on the other hand, which is at the end of the electron transport chain, is very electronegative and wants that electron. So that's sort of how you can think of it flowing down the electron transport chain. So we started with NADH and FADH2. And so why do we need an oxygen at the end of the electron transport chain? Yeah, it's electronegative. So another way to think about it too is if you didn't have an oxygen, then your electron would just end at the end of the electron transport chain and it wouldn't go anywhere, and they would just start to accumulate, and then you'd have a lot of negative charges there, and then it would stop working. But when we have this oxygen, now we can just give the electron to that, and it can just leave the mitochondria, and now you're not accumulating anything there.
They're pulling hydrogens on it.
Yeah, it's also taking some of the hydrogens that float down. Great, good point. But so you do want to conserve some things, like in the citric acid cycle, we wanted to conserve the carbon, but some things you want to get rid of as waste. So you want to get rid of those electrons out of the mitochondria, otherwise they're going to accumulate, you're going to have a lot of negative charges, and then the cycle is going to have to stop. So this makes everything a little more sustainable. What does oxidative phosphorylation mean? Anyone want to raise their hand for the first? Sorry. Does anyone else want to share in the discussion?
Anyone else?
How can you turn that down?
I know, it's difficult.
She said call him.
Doesn't
All
look
right,
like it.
go for it,
We
please.
almost did like 5 heartbeats.
Alright, so, oxidation means the loss of electrons.
Great.
And whatever that word is. It
Phosphorylation.
means adding a phosphate to ADP to get ATP.
Great, exactly. So NADH and FADH2 are being oxidized, they're losing electrons, and ADP is being phosphorylated to become ATP, which was our end goal. So why did we create a hydrogen ion gradient in this process? I hope not. What did we use the hydrogen gradient for? Proton gradient.
To move it down.
So it could flow. No,
Against.
it could what?
Flow against the protons.
So we pumped it initially up the gradient, and then we let it flow down to do what?
Oh, to the reduction
To
oxidation
consume all
reaction.
of the NADH and FADH.
No, so we let the protons flow through the ATP synthase to make ATP. So once we had this really big proton gradient, it can release energy as it flows through the ATP synthase, and that energy can be used to make ATP.
So the hydrogen ion gradient allow the electrons to flow.
No, the proton flow to make the ATP. Yeah. So the proton gradient is sort of the step after the electron transport chain. Yeah. All right, that's everything I wanted to cover today. Thank you for participating.
Like the vegetable.